All right, so our next speaker codes, but he only codes to death metal. Please welcome Paul.
So the time it took, thank you for having me, organizers. I really appreciate it. This is more of an academic thing when you're speaking to a room full of medical doctors, and conflict of interest is a big thing. I only have one financial disclosure, and that's I'm founder and owner of a data science recruiting company called TalentSeek, but that's not relevant here because none of this work or my clients had anything to do with this presentation. Okay, with that said, the time it took for me to get up here, fiddle with the display, get the PowerPoint fixed, somebody just died from cancer. It's about one person a minute, or about 1,500 roughly every single day, right? People are coming down with cancer from many different sites of their body, and I'm hoping to leave you with this idea that cancer, like many diseases, is a disease of numbers, and with those numbers comes data, and data that we can mine and try to better understand cancer. So for example, 3.2 billion is how many base pairs or sets of genetic letters that we have in the human genome. These letters are organized into about 20,000 genes, and this is kind of misleading because those genes can actually be combined in different ways to give rise to anywhere from 250 to 1 million different proteins, right? These are like the workhorses of our cells. Our DNA is about 99.6% similar to one another's, but that remaining 0.4%, that's about 12 million base pairs, right? This is where side effects from medication come. This is where predisposition to certain types of cancer comes from, and it's in those differences. And we're getting smaller here, right? So 6, so how many feet long the DNA for one of your cells would be if you uncoiled it and put it end to end, right? But that's just one cell. Some estimates we have trillions of cells, so if you put all of them together, that chain would be about 67 billion miles long. Where does all this fit, right? We have these little cells. Well, this is in a cell. And if you were to take a human hair, slice it lengthwise 10 times, that one slice would be about as big around as the nucleus. And even smaller yet is the error rate for duplicating this DNA. So these letters, as your cells divide. Now this error rate doesn't seem like a big deal until you consider all what I just told you and how much DNA each cell has, right? So that's actually about 120,000 mistakes every time a cell divides, and they're dividing all of the time. If you have a mistake in these letters, right, it can lead to a very different message. So you have the innocuous statement, oh, let's eat, Grandma, or something a little bit more grim, like, let's eat Grandma, right? So punctuation matters, like the letters, these contexts, it really matters. Most of the time, these mistakes are harmless, and we have cellular machinery that actually catches them and can actually repair these mistakes, but not all mistakes are caught. And even if we have one mistake, you know, it's perfectly fine. But a second mistake, a third mistake, eventually there's a snowballing, and eventually you end up with cancer. If you can actually see, my mouse is kind of hard to see. You end up with cancer. So in order to speak about this, we have to talk a little bit about the central dogma of biology. I've already mentioned cells and genes and proteins. The DNA is our blueprint, right? So these are nucleotides, and they make up what I like to think of as like the cookbook or the blueprint. So DNA is like the cookbook. We have these other molecules called RNA, and that's like the recipe. So you open up the cookbook, you flip to a certain page and say, oh man, I really feel like vegan pizza tonight, so I'm going to find that recipe. And then when you actually go, you bake it in the oven and you cook it, that's kind of an analogy for the protein. So you have this blueprint, your cells make a message from this blueprint, and then they create related proteins that actually carry out the actions. In this case, it's in our analogy, it's the dish. There's a variety of different technologies for measuring the mistakes of the cookbook, the recipe, the dish, or even, I don't know, the metabolites, which is more like kind of like what you sprinkle on top of, right? The accoutrement. All these different technologies give rise to lots and lots and lots and lots of data, particularly big molecular data. So a lot of times people in healthcare, they think of big data, EHR. I know somebody who's working on millions of patient records right at this moment. I work on the molecular side. And the molecular side, the clinical side, is also important too, right? All of this data is like the big data of cancer. It's important because it can tell us things about the treatments that work best for people, right? But everybody is a little bit different, as I mentioned before, so not all treatments, even if it appears to be the same kind of cancer, is going to work in the same way. Identifying folks for clinical trials, even developing new treatments or a cure. This is a bit of an old infographic. I think it's from 2015, but it's a great point too, where it talks about some of how much money goes into the cancer research, and also that most of these cancer treatments do not work. I'm a bioinformatician, or really it's just a fancy word for a biological data scientist. So I try to make sense, and my research program at the Moffitt Cancer Center is making sense of big molecular data. I sit at the intersection of a bunch of different fields, as you can see here, and in many ways we're kind of jacks of all trades, or we're like kind of a mile wide, inch deep in some cases. And so I have a mathematics and a chemistry background, right? So I'm kind of well suited to sit at the intersection of all these different fields, but I am not like a super expert on machine learning or even like computer science. But I do understand the biology, and I'm dangerous enough with my code where we can use data science approaches to try to understand cancer. This brings us to the systems approach. So many labs, even at Moffitt, they make their whole careers out of studying a single molecule, a single gene, a single protein. Generally, as bioinformaticians, that's not what we do. We're interested in the systems level. So not just one gene in a patient, what's happening with all of the genes, and how are they working together? Us to understanding the two main goals of cancer research. So if we understand what's going on in a patient's cells, we're able to say, compare someone who's sick to someone who's healthy, and try to understand, okay, well, what is different about them? Why did they get cancer? What are these mutations that are giving rise to the cancer? If we can know that, then we can develop new treatments for cancer. Treatments for cancer historically have tended to be very bad, right? So you have chemotherapy, for example, where it's a poison, and you take it, or a patient is infused, right, intravenously. They're infused with it. Their hair falls out. They get very sick. You're basically poisoning your body. You're poisoning the cancer a little bit more because these are rapidly dividing cells. They're a little bit more susceptible to chemotherapy, but you're still hurting the patient. What if we can understand what is making this tumor tick based on that simple comparison that I gave you? Can we find a silver bullet such that we can treat that specific molecule, that specific condition within the tumor, but leave the patient's body otherwise untouched? So finding new drug targets is one of the main goals. The other is early detection biomarkers, right? So for a lot of cancers, even to this day, there are no biomarkers, right? So you go to the doctor for your annual checkup. They may draw some blood, you pee in a cup, and they're looking for elevated glucose levels and cholesterol. These are biomarkers for how healthy you are. For the most part, there are no biomarkers for cancer. So you can't just go pee in a cup and the doctor says, oh, hey, this looks a little off. We should do some more testing. For the most part, that does not exist. And the earlier that you catch cancer, the better the patient does, right? So regardless of where it originates. So using these tools that we have at this intersection of this Venn diagram, and using systems approaches to try to get to new drug targets or early detection biomarkers. In a paper that we published now a couple of years ago, we were trying to understand squamous cell lung cancer. Squamous cell lung cancer is the second largest subtype of non-small cell lung cancer, right? So when you talk about lung cancer, there's actually a variety of different types of cancer that you can get. Squamous cell lung cancer in particular has a fairly high rate compared to some of the other cancers, and it is traditionally thought of as a smoking-associated disease. If you sort of lose the genetic lottery and or you're a heavy smoker and you get squamous cell lung cancer, you have limited treatment options. For some patients where they develop cancer, there's something called a driver mutation, right? So that error that I was talking about before in the DNA, it occurs a lot. So there are common mutations that show up in a lot of patients. Doctors and researchers have figured out that some of those make a protein that you can treat with a drug. But these patients with this particular mutation that makes a particular mutated protein is the vast minority of cases, right? So again, we need to understand this disease better in order to get to better treatment, better early detection biomarkers. So people knew about squamous cell lung cancer. They even had some idea about the molecular underpinnings of this disease. But we wanted to do a systems-level, multi-omic approach to profile these tumors and really get to what is going on with them and to understand them such that we can get to treatments and biomarkers. This is the cohort that we used. It was predominantly older, predominantly Caucasian smokers. And on the right-hand side here is a little bit about the methodology and the experiments that we did. And then so we take, at Moffitt, right, we're a research hospital, we're a research institute and a hospital. So the patients that come in, they are most of the time consented, saying, hey, if we take your tumor out, can we please keep it and use it for research? So we identified 108 cases of squamous cell lung cancer. We took, essentially the surgeon takes this out of the patient and they just drop it into liquid nitrogen to preserve it such that it is preserved and we can look at things like DNA and RNA and proteins. The pathologist also needs to look at it, so they also take a piece of this tumor, take a microscopically thin slice of it, put it under a microscope, so that way the pathologist can look at it and say, oh, this is stage 1 or stage 2, or it's this kind of cancer, et cetera. And then so with this cohort, we subjected them to targeted DNA sequencing, RNA sequencing, a copy number array analysis or copy number variation analysis, which is actually counting how many genes. You can get extra copies of genes, which also can drive cancer. RNA sequencing, and also proteomics, which is what my lab specializes in. So this is looking at large-scale protein expression in tumors, right? You can look at DNA, you can look at RNA, you can look at protein. My lab specializes a lot in the protein, but then also systems level, how does all of this interact and work together? Oh, and I already kind of, it would have been a nicer figure, I guess, to have up here. But yeah, so we're doing a systems approach where we're going to take the DNA from these patients, take the RNA from these patients, take the protein from these patients, excuse me, their tumors specifically, and profile them to understand the interplay and can we get to new treatments, early detection biomarkers. So a big problem in cancer research is just like, how on earth do we classify these patients, right? We have some patients, we have some data associated with them. Maybe we've extracted DNA or RNA or protein, but like, what does it mean and how does it fit together? So one thing that we can do is we can just cluster our data. I mentioned we did proteomics experiments, right? This is large-scale global characterization of the proteins that are in tumors. These experiments identify over 8,000 proteins, and it's across 108 patients. Well, that's a matrix, right? We've got a matrix of features. We have columns of samples, or tumors in this case. We can do something like consensus clustering, which is just a cluster of clusters approach to try to get at what are my inherent groupings within this cohort of 108 samples. Because I don't know what they are. I know most are stage 1, some are stage 2, some are stage 3, they're mostly white. But at the molecular level, what does this look like? And what can this tell us about treatment or early detection biomarkers? So we can do things like consensus clustering and picking a K, right? So everybody, it's kind of hard to pick, well, if we're going to do supervised clustering, what K do we pick? And then so you have metrics that you can apply here, just like you can with most data, to understand, well, what are my groupings? What are my numbers of clusters? And then so with our protein data, right, we have lots of data. With the protein level data, we're going to cluster them. We have some of these metrics where we can say, oh yeah, kind of, the graph kind of bottoms out around 5. This looks like it's converging. We can call it 5 clusters. And we can look at the tracking plot. We can look at the group membership, 4 different Ks for a particular patient sample, and understand, is its classification, is its cluster relatively stable relative to the others, or is it moving around a lot? Because if it's moving around a lot and it's not stable, maybe that's not a great K to pick. So we take this and we say, okay, it kind of looks like 5 groups, but this is just the protein data, right? So we're doing a systems level approach. We're going to integrate this with all of the other data that I had, and it leads to something like this. And I like to refer to this as the $1 million figure, right? Not only because it's kind of pretty, but it also cost $1 million, right? Between all of the processing and paying for the surgeon, paying for the sample collection, paying for the DNA extraction, the RNA, the protein, having the analysts, I led the work on this, putting it all together, figuring out what it means, it's like $1 million, right? Our findings were that, at least here, I'm going to kind of hand wave and summarize a bit, but those 5 clusters, actually 2 of them and 2 of them actually seemed pretty similar. So you can see the green bar across the top. We identified 3 subtypes of squamous cell lung cancer based on clustering. And then we can, all right, so this is a protein level heat map where you can see the grouping on top. But then we can overlay all this other data that we have to annotate these samples and really understand what is going on with them. And okay, we cluster them and they behave one way, but what mutations do they have? What does the RNA look like? And we can do multi-omic, multi-layered experiments or analyses to try to interrogate and understand the data. One particular finding was this inflamed subtype. And we wanted to understand what is going on inside of these tumors. These tumors were inflamed, right? So like they're irritated. They're immune rich. So based on the signals from the protein, the signals from the RNA, lots of signals that we're receiving in all of the data, it says that these samples are immune rich. But there's, right, thousands and thousands of proteins. Some are going up, some are going down. How do we make sense of all of this data? I'm no expert. I don't have all 20,000 genes or proteins memorized, right? So we have these tools called pathway enrichment tools, or we can perform pathway enrichment analysis, where we take a big, huge list of features, maybe 1,000 different proteins that are elevated in this inflamed subtype that we've identified, and we can condense them down into only a couple of pathways, right? So this is a consolidation of information, dimension reduction, and we go from a whole huge list of features, in this case proteins, to a much smaller list of pathways, or the biological processes that this cancer cell is using. So when we do a pathway enrichment of the inflamed subtype, we see on the right-hand side here, these blue bars, all right, there's a adjusted p-value on the x-axis. But this is shouting out to us, hey, these are inflamed tumors. There is increased immune response from these different pathways. So each one of these bars is a different pathway, which has some proteins inside of it, right? We're consolidating the information together. And so this is screaming immune. But there's a problem, right? So I mentioned that we took all these tumors, we ground them up. We're actually don't know exactly what's inside of the tumor. There's going to be some cancer cells, but there's also going to be a whole bunch of immune cells. These immune cells are critically important because there are new immunotherapies that are available that essentially take the brakes off of your immune system and allow you to treat a patient, and then their own immune system can attack the cancer. So a fundamental question that we have is, well, what is inside of this tumor? What immune cells make it up? Because that's critically important to immunotherapies and these treatments. So some clever person said, hey, well, if we have this tumor and it's a whole mixture of cells, we can actually just model this. If we just assume this tumor, right, it's some cancer cells and some immune cells and some endothelial cells, like blood vessel cells. So we have a whole bunch of signals that are summing up to this mixture. If we can go independently isolate some cancer cells and some T cells and some B cells, some various immune cells, endothelial cells, we can derive a signature. So then the mixture on the left is what we've measured. That is known. The signature is measured. Now that is known. The only thing that we don't know is how these are combining to make the mixture. It's a linear algebra problem, right? So we have these immune deconvolution tools, which given some sequencing data, we can throw it into the algorithm and get back out how many cells, or what are the relative populations of some of these cells. And that looks like this. So we've just done a simple box plot. I'm using nonparametric statistics to compare the levels of these scores that we're getting out of the deconvolution algorithm. And again, all these different immune cells that we have, they're elevated, or at least the neutrophils, the memory B cells, the monocytes were all elevated. And we can, I'm going to skip ahead a little bit. We can actually see this. So if, right, we've been just grinding up these tumors, I was telling you, we've lost that resolution. But we have samples left over. We can go back to these patients, cut new slides, put them under a microscope, and stain them, and ask the question, do we see these immune cells that all of our omics technologies are pointing to? So I've circled some regions here that are essentially immune hot, and what we can do is go, and this is a very zoomed in picture of some of these immune structures that we observed in the cells. So these are cells underneath the microscope inside of a tumor that have been stained with immunofluorescent tags to light up the different populations of immune cells. And recognize the problem here. It's like, I'm just grinding all these up together, and then when I measure it, it's just everything kind of mixed together. There's spatial data here. These individual cells are relating to one another. The next layer, sort of the next big thing in the work that I do, are spatial analyses and single cell analyses. So instead of looking at the tumor as a whole, now you're getting down to the individual level. But then it's like an exponentially increasing, exponentially more data. Real quick plug, because I'm out of time. Moffitt Cancer Center, we run a biodata club. We have funding through the R Consortium, and it's kind of like a monthly R meetup, but we also talk about a bunch of different languages and libraries. And this was very much billed as, we get a lot of scientific talks at Moffitt, and it's just like, I don't want to see another talk. I want to actually know, like, how do you run these packages that you're talking about? Or like, what goes into doing some of these analyses? And it's got a heavy, like, molecular, heavy health informatics slant to it, but all are welcome, and we have a meetup group if you're interested, and we're going to have a Cancer Hackathon in February. And these are just acknowledgement slides. So I have funding through the Cancer Research Institute, which is a foundation that's funding some of those pretty pictures that I just showed you. And I've had funding from the National Institutes of Health, from Moffitt, our department funds pilot projects, and others. Thank you so much.